Hello everyone, my name is Maya George and today we'll be discussing the findings of the article A Dual Mechanism Antibiotic That Kills Gram-Negative Bacteria and Avoids Drug Resistance by Martin II al. Antibiotics are drugs used to treat bacterial infections. During the golden era of discovery, over 20 classes of antibiotics were developed. Each class has a specific mechanism of action that makes it unique in how it is able to kill bacteria. For the purpose of this presentation, there's a few I'd like to highlight, which will be discussed further. Firstly, we have antibiotics that target folate synthesis, such as trimethoprim and sulfonamides. These compounds target enzymes in the folate pathway, which is critical for the creation of nucleic acids in bacteria. Next, we have antibiotics that target the cell membrane integrity, such as polymyxins and lipopeptides. While the golden era revolutionized science, after the discovery of all of these antibiotics, we have now reached a point at which antimicrobial discovery is limited. In addition to this, the prolonged use of antibiotics has resulted in many strains of bacteria developing resistance, so that many antibiotics are no longer effective. This has created a global health crisis in which there's a drastic need to discover new antibiotics. In order to discover antibiotics with new mechanisms of action, researchers at Princeton University conducted a screen of approximately 33,000 compounds in search of compounds inhibiting the growth of E. coli IPT D4213. They specifically chose the strain as it has a compromised outer membrane, making it partially permeable to compounds targeting both gram-negative and gram-positive bacteria. Of this screen, one of the most potent hits was SCH-79797, a previously reported PAR1 antagonist. An ideal antibiotic is hard to develop resistance against, kills both gram-negative and gram-positive bacteria, presents limited toxicity to the host, and is able to penetrate both infected host and bacterial cells. Therefore, researchers conducted a series of tests to determine if SCH-79797 fit this criteria. During the course of their experiment, researchers assessed bacterial susceptibility, in vivo activity, resistance, determined the mechanism of action, targets, and created derivatives of the compound, and these were their findings. To assess bacteria susceptible to the compound, researchers measured the MIC against various clinically relevant pathogens. After incubating the bacteria for 14 hours at 37 degrees, it was found that SCH-79797 significantly hindered the growth of multiple gram-negative and gram-positive bacteria, including Neisseria gonorrhea and MRSA, with no signs of resistance. Next, researchers sought to determine if the compound could act as an effective antibiotic in vivo. They infected the waxworm Galleria molinella with a lethal dose of A-Baumannii, and then treated it using SCH-79797. The result was a survival rate of Galleria molinella similar to positive control antibiotics. They next sought to determine the frequency at which bacteria could develop resistance in the discovered compound. Over the course of 25 days, they passaged MRSA and A-Baumannii in sublethal concentrations of SCH-79797 and used novobiocin, trimethoprim, and niacin as control groups. While they isolated resistant mutants from all the control groups, no SCH-79797 resistant mutants were found. Upon this discovery, researchers sought to determine the mechanism of action. Using bacterial cytological profiling to compare the phenotypic death states of the bacteria, they found SCH-79797 had a death phenotype unlike any of the other antibiotics tested, further indicating the idea of a novel mechanism of action. To determine the targets of this compound, researchers used thermal profiling, CRISPR-I, and enzymatic assays. A thermal profiling proteome assay was used to compare the thermal stability of the entire proteome. When treated with SCH-79797, it was found that the thermal stability of DHFR was shifted. These results were consistent throughout the experiments, indicating this shift can be attributed to the binding by SCH-79797. Using CRISPR-I, it was found that when genes upstream of DHFR were knocked down, Bacillus subtilis was hypersensitized to sublethal doses of the antibiotic, indicating that a change in the folate pathway influences antibiotic activity. In an enzymatic assay, it was found that following the treatment with SCH-79797, levels of dihydrofolate, the substrate of DHFR, increased tenfold compared to untreated cells, and metabolites downstream of DHF dropped significantly, which is a characteristic indicator of DHFR inhibition. Trimethoprim, a known inhibitor of DHFR, was used as a positive control in each of these experiments and produced the same results. So based on these results, it was concluded that SCH-79797 inhibits DHFR. To find the second target of SCH-79797, researchers used flow cytometry to detect changes in the characteristics of the cells. After the treatment of E. coli IPT D4213 with SCH-79797, For 15 minutes, staining revealed significant defects in membrane permeability and polarization. This confirmed that SCH-79797 not only competitively inhibits DHFR, but also independently disrupts both membrane potential and permeability. Upon these findings, researchers sought to determine whether it was more effective to have multiple mechanisms of action in a single scaffold or co-treatment with multiple antibiotics. They tested SCH-79797 against combinations of trimethoprim, niacin, and daptomycin on MRSA USA 300 persister cells. It was found that SCH-79797 was able to kill these cells while combination treatments could not. Furthermore, when combinations of trimethoprim, niacin, and polymyxin B were compared to SCH-79797, it was found that after 2 hours of treatment, the co-treatment methods had a greater amount of viable cells due to antagonist activity of the combined antibiotics. This brought them to the conclusion that combining multiple mechanisms of action into a single chemical scaffold is more effective than co-treatment. Lastly, to link the antibiotic activity to specific areas of the scaffold, researchers created two derivatives of SCH-79797, irresistant 10 and irresistant 16. For irresistant 10, they removed the isopropyl and cyclopropyl groups. This compound was a more potent inhibitor of FOLA than SCH-79797 and did not target the cell membrane. For irresistant 16, they replaced the isopropyl group with a biphenyl group to increase membrane targeting activity and removed the cyclopropyl group to enhance DHFR inhibition. This derivative had more potent activity than irresistant 10, indicating that targeting both membrane integrity and folate biosynthesis is more powerful than targeting folate metabolism alone. In addition, it had an increased antibiotic activity without increasing mammalian toxicity, and a 100-fold increase in therapeutic potential compared to SCH-79797. It was found to be extremely stable in vivo, showed robust in vitro activity towards N-gonorrhea with an MIC of 0.03 micrograms per milliliter, and in a mouse model was found to significantly reduce the vaginal load of N-gonorrhea. Overall, these findings are a significant contribution to the field of antimicrobial discovery. They indicate that combining multiple mechanisms of action into a single chemical scaffold may be a vital approach to targeting challenging bacterial pathogens with limited resistance. In addition, they detail a strategy to discover antibiotics with new mechanisms of action. Some of the pros of this paper is that they had a very thorough lab workflow. They checked the structures for mammalian toxicity, activity in vitro and in vivo, the ability to target gram-positive and gram-negative bacteria, and the possibility to develop resistance, which are all crucial aspects of discovering an ideal antibiotic. However, some of the cons are that a limited range of antibiotics were used to test the effectiveness of combination therapy versus SCH-79797. It is possible that different antibiotics may exhibit synergistic or additive effects, unlike the compounds tested. In addition, a broader range of bacteria should have been used to prove the antibiotic is effective against resistance. For example, using environmental isolates as well as clinical isolates ensures that the tests are representative of the general population of antibiotic resistance genes. Once tested against a variety of bacteria, an MIC 90 would indicate a true resistance-free antibiotic. Some suggestions for next steps are to test SCH-79797 against relevant resistance genes such as those found in the CAR database, to test potential resistance in both environmental and clinical bacterial isolates, to create linker compounds with niacin and trimethoprim and polymyxin B and trimethoprim to confirm the idea that combining multiple mechanisms of action into a single chemical scaffold is the ideal approach, and to use different antibiotics targeting folate synthesis such as sulfonamides in the bacterial membrane to confirm that SCH-79797 demonstrates more potent activities than combination therapy. To end this presentation, I would like to thank my PI, Dr. Jerry Wright, the course instructors, Dr. McDonald and Dr. Sloboda, our TAs, and all of my classmates. Thank you.